Thank you. So for those of you who don't know me, I'm Bill Beck. Recently retired, but I was Dr. Wynn's predecessor at the Cancer Center for about 15 years before. And I have to say there is life beyond the Cancer Center, as I spent more than 17 years in administrative positions in the College of Pharmacy. And before, but this isn't about me, before introducing Dr. Dalton, I'd like to thank Dr. Wynn for his outstanding leadership in the Cancer Center for these past 5 or so years, and to wish him all success in his new position as Director of the Cancer Center at UVA. But this isn't about Dr. Wynn either. So I'm honored to be able to introduce my friend of long standing, Dr. Bill Dalton. I've known Bill since he was a fellow. That's almost before electricity. And we've stayed in touch for the better part of almost 40 years, and we've actually published a few papers together. Bill received his undergraduate training in New Mexico, did his PhD in pharmacology and toxicology, and his MD, dual degree program at Indiana University. He did residency and oncology fellowship training at the University of Arizona, and he's board certified in internal medicine and oncology. And he stayed in Arizona to eventually become professor of medicine and deputy director of the Arizona Cancer Center. Prior to taking his present position at M2Gen, more of that in a minute, Dr. Dalton was president and CEO of the Moffitt Cancer Center, NCI Comprehensive Cancer Center. And before that, he was dean of the University of Arizona School of Medicine. I'm sure he's got a lot of stories about that. We were just talking about some. And before that, he was Associate Vice President for Health Sciences at the University of South Florida and a leader in the Moffitt Cancer Center. It was there that he set up the unique Department of Interdisciplinary Oncology. Bill is the founder, executive chair, and I think now former CEO of M2Gen, a subsidiary, or maybe former subsidiary recently, you'll straighten it out, of the Moffitt Cancer Center that's has developed an approach to follow cancer patients throughout their lifetimes, a concept that Bill developed called total cancer care. This TCC concept is part of precision cancer medicine. It's been adopted by 20 academic cancer centers in an alliance called Orion, which Bill will talk about. Orion is an acronym for Oncology Research Information Exchange Network, for which Bill is a major driving force and co-chair, and he'll be telling us about these efforts. He's well known and well regarded nationally and internationally, has over 200 publications, served on many advisory boards, including the National Cancer Advisory Board for the National Cancer Institute, or the Board of Scientific Advisors, I should say, and many, many more, and he also holds many patents. So to summarize briefly, Dr. Dalton is a physician scientist, internationally known for his laboratory work on drug resistance, tumor microenvironment in multiple myeloma, and more recently developing information systems, Orion, that allows sharing of patient data to predict which patients will benefit from a particular clinical trial based on their genotypes and phenotypes. For his leadership in the area of personalized medicine, he was recognized as the 2010 recipient of the Personalized Medicine Coalition's National Leadership in Personalized Medicine Award. And earlier this year, I'm very pleased to say he was also elected a Fellow of the American Association for the Advancement of Science, the AAAS. So without further ado, please join me in welcoming Dr. Bill Dalton back to you guys.
Well, I've been looking forward to this day for some months, and I want to thank Dr. Wynn and my good friend Bill Beck for inviting me here to speak to you. As I said, I was looking forward to it, and I'm not disappointed. What I see here is frankly tremendous. I enjoy talking to you and the people I've met. It's palpable enthusiasm that I see and commitment. So I just wanted you to know that. So it's a thrill to be here. Thank you very much. So, as Bill said, what I'm going to talk to you today about is an approach to following patients in partnership with patients to learn from each patient. And by learning, we can actually generate evidence of value. In other words, what's therapy working for whom? And without that evidence, we're flying in the dark. And the only way we'll ever gain evidence to understand what is working, what's not working for whom, is to ask patients permission to study them and follow them. And that's critical. We can't just do it. We must engage the patients. And so if there's anything I hope you leave with, it's start with the patient, ask their permission, and then create a partnership so that we can all learn. That is the essence of what we're going to be talking about. Now, this is also a way, you know, I got my MD at the age of Hippocrates, okay? But it was reactive. The patient comes in, sees the doc does an exam. This is even back to Hippocrates. And the physician reacts to the scenario they're seeing. That was good then, but it's not good enough now. We're, number one, living longer, and frankly, we're getting more diseases. So what if, by learning from patients, we could actually, if you will, assign them to a community that looks like them, and then be able to predict what they are risk for and what they will need. So what I'm about to describe is a partnership to identify need for patients so that we can anticipate their needs. What if we could, by that anticipation, actually intervene before the need manifests? So being able to predict by learning, using data science, going from reactive to proactive medicine. Thank you very much. I do have a few sides to do this. As Bill said, I am currently the chair and founder of M2Gen, which was a wholly owned subsidiary of the Moffitt Cancer Center, now owned also by Ohio State, which is one of our key partners in forming Orion, and now more recently the Hearst Family Information Company. I'm sure you've all heard of Hearst. So those are my... So we've been talking about personalized medicine for a long time. The sequencing of the human genome, I believe in 2003, was exciting, and we're still talking about it. What we need to do is interpret this ability and translate it into value, again, and evidence of value. So we're on our way, but here are the questions that I would pose to you, because this is a societal effort. This is not something any one cancer center can do or any one industry can do. This takes, what we're embarking on is a societal issue. It's going to take society coming together and saying, this is what we want to do in healthcare. I'm going to focus on cancer because that's what we do. But I would like to think that everything we're saying in cancer could be done for every potential healthcare issue, including preventive medicine. So how do we create a system that identifies individual patient need? We can only do that by, again, asking their permission. And we develop markers that would actually predict that need and risks. What if we could, again, intervene before the need manifests? Wouldn't that be grand? How do we develop these? It's a genotype-phenotype combination. There are many factors that influence whatever health malady a patient may have. So we need to include them. And of course, our genetics is an important part of that. So how do we use that, again, to predict? You're going to hear me talk about predict, anticipate, evidence when we treat. And obviously, by doing so, raise the standard of care for everybody at a reduced cost. Now, that's a real challenge as well. Yeah, it does, doesn't it? I have a plan, though. Anyway, I was quoting someone else. In this case, you may know that America pays more of its GDPR than any other country in the world. 20% of our gross national product goes to healthcare. The closest to us is 10%. And by the way, our quality of care in terms of survival is 34th in the world. We're paying twice as much, and we're not even close to the best. That is the brutal information that we have to acknowledge, which should tell us what we're doing is not good enough. All right? And so, with that recognition, how do we get there? I will submit to you that forget about it won't work. So we have to come up with something. And it's going to, I thought I would never do this, but I'm, as a physician and a scientist, going to talk to you about a business theory. And this is a paper I read in 2016 in the Harvard Business Review. I was actually trying to study networks and what makes a network work and one that doesn't. And I found this paper by Mark Kramer and Mark Pfitzer. And what struck me was the title, Creating an Ecosystem of Shared Value for Healthcare Through Collective Impact. Basically, what they were saying is, what if industry and other stakeholders came together, not about healthcare, this is about any business, would come together and create a resource that no one stakeholder could make. And each business benefit from the creation of a resource that required collective collaboration, impact. So, and that this ability to work together, even though they may be competitors in the business realm, create a resource that improves society. That's the message of this paper, and I would recommend everybody to read it. Again, you'll find it in the Harvard Business Review in October 2016. But I read this and I thought, this is what we're trying to do. We're trying to create a better ecosystem to deliver evidence-based medicine, but no one industry stakeholder can do this by themselves. But they came across, which I found fascinating, 5 elements that were required to make this ecosystem work. The first was a common agenda. All stakeholders involved have to agree what they're trying to build. If you don't agree with what you're trying to build, it will never work. So that's number 1, a common agenda. And I would submit to you in healthcare, it's developing a data system that follows patients so that we can learn and then treat patients based on what we've already learned and improve the evidence, which should reduce the cost. So that's the resource that collectively we should be aspiring for. So that's the common agenda. Build a resource that creates evidence of value and supports discovery, supports translation of those discoveries, and supports delivery of evidence-based medicine. And you have to have shared measurements. Part of the business aspect of, if you can't measure it, you're not going to succeed. You have to, as you develop this agenda, create metrics of what does success look like. Define success and measure it. Otherwise, you're fooling around, okay? You're not going to get there. Mutually reinforcing activities. This means while you may be working with other stakeholders who you actually compete against, you understand the value of this new resource, how it can help you, but you have to understand why these other stakeholders are there and what are their incentives in creating this new resource. So, and a mutually understanding of that. Constant communication. Man, is that isn't true. Constant and in any form, but you can't have stakeholders doing their own thing, thinking they're contributing to a new resource and expect this to work. Everybody has to talk about why they're doing what they're doing, all right? A dedicated backbone. Frankly, as you'll see in a moment, this Orion effort is that ecosystem we're trying to build in partnership with patients and other stakeholders, but as you'll see in this paper, you need a backbone, a foundation of the engine that is behind it. And as I hope you'll see in a minute, this company that Moffitt formed, M2Gen, is that engine. If we required every stakeholder to build their own foundation, it would never work, ever, because it's not mutual. And frankly, it would be too expensive for everybody. And then, again, those 5 elements. Equally important, perhaps even more important, are who are you partnering with? Patients are at the top. That's why we're here. Everybody in this room, you're here because you want to help patients. And we know that our system now is falling behind. in that ability. But these are the other stakeholders that we need to work with to create this ecosystem and a resource that we all aspire to. Data that demonstrates evidence supports discovery, translation, and delivery. And so you can see these are the ones that we consider now as stakeholders in this overall effort. But patients are paramount. As I said before, unless we ask patients permission, it won't go. When we do ask their permission and they participate, the strongest voice in accomplishing this are the patients. I can talk till I become totally hoarse, but a patient, when they say, we want you to do this, everybody listens, all right? So this is the alliance of Orion members. We're now at 20 members. It started 5 years ago between Moffitt and Ohio State. And here you see, we're coast to coast. We do serve, I think, quite a diverse group. That happens when you're coast to coast. For example, Murtha Cancer Center is the cancer center for the active military at Walter Reed, have joined us, and working with the Veterans Administration as well, following patients in active military throughout their retirement as a veteran. They're part of it, a keen part of it. Morehouse School of Medicine, traditional African American school, very contributory. University of New Mexico. In July, I was meeting with 12 governors of the Pueblo Native nations. They want to participate, but on their rules, which I don't blame them. There has to be a trust factor here. But I think you can see the goal is to make this accessible and inclusive, which brings us to our guiding principles for Orion. Number one, we're doing this to collaborate, because by collaborating requires sharing, in this case data, but that sharing data is not enough. Sharing data is the first step toward collaborative learning. If we all agreed to share, but we stayed in our silos, you don't accelerate learning that way. You do that by working together, so collaboration. Inclusiveness. It's our belief that every patient everywhere should have access to this. If we believe we're creating something of value for patients, then every patient, it's our obligation to see inclusiveness here. And I'll be very frank, I get excited when I talk to you folks here. You guys get this message in spades. I'm very pleased about it. Accessibility of data. If we put it in silos, it's not there, and the collaboration is gone. And partnerships. And that partnership is not only with those stakeholders, but with key other private-public partnerships. Partnerships with the regulators, i.e. government. Partners with industry, i.e. pharmaceutical companies and biotech. They're all important, and they need to be at the table, and they need to acknowledge everybody's incentives. So, the backbone of this effort is a protocol. It's an observational study, IRB approved. It's in its 14th year. As Bill said, we call it Total Cancer Care Protocol. The idea was following patients throughout their lifetime with their permission. They're donating their data and specimens for us to study longitudinally, i.e. throughout their lifetime. Then they can withdraw their consent anytime they want because they know they're in our system, which is very, very important. And because they've consented, we can contact them. You can actually ask, how are you doing? Now, if they don't know they're in a system and someone says, ask them how they're doing, well, I'm the first thing, well, who are you? And why are you asking me? In this case, they know who we are because they're part of it. And that to me is critical. And the other thing, what if we found something of benefit for those patients? You say, okay, we know that your disease has progressed, you need a clinical trial, here are the clinical trials. Again, if you cold called someone and under those circumstances, that'd be a turnoff, lack of trust. In ours, they know they've given us permission to contact them if we discover something that they might benefit from, such as a clinical trial. So I can't emphasize enough the importance of starts with the patient, starts with their consent, and constant communication. Again, what started at Moffitt 14 years ago, we now this slide's already out of date, we have 20 with over 300,000 patients enrolled over that time. And with the Alliance as it stands now, we're enrolling approximately 500 patients per week. So it's a good start. Here are the goals, if you will. I think they're fairly straightforward. Data science to accelerate discovery through data sharing. And again, data sharing is step one. Step two is work together. collaborative learning. We think that the greatest need that we are serving today for patients are those patients at risk for progression on standard therapy. They need something new, and that newness is a clinical trial. So you're going to hear me continuously emphasize serving the patients in greatest need. Again, those are the patients with more aggressive disease and that will likely progress on standard of care. We need to create options for them before they need it. That's, again, anticipate. And I'm going to show you how we're doing that. And then ultimately develop a tool, a clinical decision support tool for the physician who, by the way, doesn't have a degree in genetics and genomics. So you can't expect them to understand what a phospho-AKT is going to do in the standard of care. What we need to do is present the data that this is the population your patient belongs to, and this is what's working for that population. That's what physicians want to see. And that would be the decision support tool. Now again, M2Gen is a company that was wholly owned by Moffitt, now Ohio State and Hearst, whose goal is to be, it is a for-profit company, because one thing I learned, first of all, my board told me you need to make it for-profit for two reasons. One, we could get economic development money from the county and state. Jeb Bush gave us $10 million. And other industries want to hold other industry partners accountable. And so as a for-profit, they feel they can do that more. So it was to attract that sort of ability. But this is what it's M2Gen's job is to serve our members, and that's what the members in the Orion, that is the alliance. But we also have responsibilities to other stakeholders that are participating, which obviously includes patients, we'll talk about more in a moment. But I wanted you to understand the role of M2Gen here. Basically, it's a dedicated workforce. It's that backbone that I mentioned in the elements for ecosystem of shared value. So the protocol's been together for 14 years. I'm pleased to say that the faculty are using it. And these are just some of the publications that have come. We have well over almost 200 publications that have been supported by this database. I think, and some journals that you might recognize: Cell, Nature, Science. The quality is, I think, impressive. But again, we also work with our other stakeholders, in this case, pharma. And again, I think of the data system as being a resource for discovery, translation, and post-market analysis. It's that post-market analysis that doc in Kissimmee, Florida, who's seeing a new patient can ask, how many patients in your system look like this patient, and what do I need to know in order to make the right decision about how to treat? So we think of the use of the data system from a fairly high perspective of discovery, translate, deliver on the evidence, and do so with other stakeholders such as pharma. I'm going to just show you a highlight of this. This paper was actually published, I think, in 2012. So it's getting up there in age. But it was, I think, a very good example of using the system. The question of the investigators was, and we were following patients generating molecular data, and the question was, in melanoma, could they find a genetic expression signature that would predict response to immunotherapy? And they did. It was a 12-gene chemokine signature. But they then said, when they found this, and they validated it through pathology showing infiltrating T cells and response to therapy, the question obviously became, well, if it's working in melanoma, will it work in other diseases? And in 3 months, they found 32 other diseases that expressed the signature and also predicted response. 3 months. Now, that's a long time, the way we're using the system now, but if you tried to do that without the data system, it would have taken a decade to generate that kind of data de novo. The data had already been created. It was analyzing the data that had already been created that allowed them to take an observation in one disease and apply it to over 30 others. So that is a power that I want to illustrate. And this was done I just asked the question, take the signature, and this is just a few days ago, take the RNA sequence signature and see if it correlates with PD-L1, which is also a predictor of response. And within an hour later, they gave me this graph. So we've made some advances in rapidity. Now I'm going to come back to serving patients and serving those patients with the greatest need, those patients that are likely to progress on standard of care. What we want to do is not react, but anticipate by following these cohorts over time and learning from them who's responding, who's not responding, those that aren't responding, what expression and what other molecular variables might determine what they may ultimately respond to. But we need to anticipate, predict, and by anticipating, create options. That's the essence of what we call the avatar. And we had to come up with a cute name. We couldn't just say similar patient. But that's the essence. Define each patient, phenotypically and genotypically, and then assign them to a cohort that looks just like them. Now, this is an iterative approach because the primary cohort is at time of diagnosis prior to therapy. Then they get treated, and in that primary cohort, you get responders, non-responders. So from one cohort, now we have two, responder, non-responder. And so it's a cascade of analysis that begins with the primary avatar. Okay, that's a very important concept that we have in order to move forward. So this is basically, again, with the consent of the patient. We are collecting their data. They've donated their biospecimen. We perform, as you'll see in a moment, a very comprehensive molecular data analysis. And then we start looking for, by using the longitudinal data, who's responding, who's not responding, and if you're not responding, what therapies might be suitable for you if you progress. Okay? Anticipate. The clinical data is quite comprehensive, and this just gives you the demographics of the type of data we're collecting on every patient. This is arduous. How many people know what natural language processing is? It's a great idea. By that category, I hope it works about 25% of the time. It's still brute force. We've got people abstracting from the primary sources, putting it in a data dictionary. And it takes us a few hours on each new patient to populate the data we need. I'm all for natural language processing. I actually was part of a grant that did it. The person that ran the grant was the godfather of natural language processing, James Allen. But he was new to healthcare. He worked primarily in DoD, DARPA, and in NASA. We received a DARPA grant, and it was called Big Mechanism, using cancer data to develop new NLP rules. This person who literally discovered NLP looked at me and said, "This is the worst piece of in terms of getting data." And he was talking about electronic medical records, totally unstructured. And so that's some of the challenge we're dealing with, but we have to work together to figure out. So that, not a beautiful description of the clinical data process, but you get the idea. In the molecular profiling, far more structure, frankly, far easier to use. The health informaticists at our place, you can always tell them because they're walking down the hall with their head down. When you see a bioinformaticist, they're going, "Oh, look what I found." So this is where we are in working with the avatar patients. By the way, we do not only somatic or tumor, we're doing germline. So we're doing whole exome sequencing on both tumor as well as germline. The germline is very important because that is the basis of pharmacogenomics. It will also is the basis for genomic ethnicity, which will tell us which populations are at greater risk of progression of disease or perhaps even development of disease. So I think that's important to call out, especially with the populations that you're serving here. I think you would do marvelous things with this type of data. And then RNA-seq. When we started the Total Cancer Care Protocol in 2006, there was no RNA-seq. It was called microarray. And now we are at the capability of using, and the other thing that we had to do, everything we studied in 2006 on specimens was fresh frozen. Now 85% of the tumors we examine are FFPE, formalin fixed paraffin embedded. That is enhanced by magnitudes the ability to study patients. And so we're thrilled. The Avatar began effort about 3 years ago. We are already with over 17,000 patients enrolled. Just as a point of reference, the TCGA, which was a great study supported by the government, was a 6-year study, enrolled 11,500 patients. In half that time, we're at 17,500. And we have longitudinal clinical data. TCGA does not. So I just use that as a point of reference, okay? So we need to continue to grow it. These are the types of analytic capabilities that the team has performed and can literally do it in real time. Something we'd invite you to participate in, perhaps a website or webinar, and you give us some questions and we'll answer them using the data in real time. So I would suggest we think about that. So this is all about integration and annotation. As I just mentioned, the real struggle for everybody that's in this arena is getting the clinical data, analyzing the data with QA/QC processes. Patients' data reside in multiple primary formats. Many of them don't agree. They'll even not agree on sometimes on gender, for example. And so we need then a QA/QC process once we've extracted the data from the primary sources. That all takes time. And so we can generate molecular data in 4 weeks. 2 months later, we're still working on the clinical data. And by the way, everything I just said is true for everybody in this field. So it just tells you the work and where we need to focus. Now, so how do we define similarity? It is on clinical as well as molecular data, phenotypic and genotypic. It's primarily based on the question you're asking. Now, we are using what's called a data lake format. In other words, you collect the data as you need it and then refine it, as opposed to a data warehouse where you just dump everything in and see what you need and hope you have it. So we're using a data lake approach. But the similarities can be categorized from a very high level in 2 ways. One, in features. In other words, if you're looking for a marker or a prevalence of a marker in different diseases, that's a use of the data for a feature. Does that patient have a P53 mutation or a KRAS mutation or an EGFR or the combination of systems? We call that feature analysis, and when you're developing the data you're collecting, you're thinking, what features am I looking for to address what question? All right? The other approach is on outcomes. For patients treated with a given disease, what are their outcomes? And the reality is everything we do is a combination of both, feature and outcomes. Now, however, having said that, when we're looking, for example, for clinical trial matching, and you have a target that you want to match, so it's a target-based clinical trial. First thing we do is go to the database of 17,000 people and say, all right, how many of the 17,000 people across all these diseases have the marker? And that's where we start, okay? And so that is a feature approach first, and then you go into you're generating outcomes because you're doing a clinical trial, okay? So I just wanted to give you some background of how we approach gathering clinical data. Now, this is a very sobering slide. MyMD stands for myeloma doctor. Don't ask me anything else about anything else. So I asked some of my colleagues that I was working with, let's see how similar we are in approaching patients with myeloma, and so that we can form these cohorts so we can learn from them, right? You're looking for degree of similarity. So, and in part, it's called reinforcement learning because this is where you've heard of artificial intelligence and deep learning. In this case, it's a reward, action and reward. The action is what therapy did the physician initiate for a patient. The reward was response. The patient did better. The patient responded. Their outcomes were improved. So that is what reinforcement learning is designed to do. Here is an action. Here's the reward. So we came at it, and this is called a Sankey plot. So a Sankey plot just plots out over time the different interventions for each population or each patient. What you'll see on the far left is newly diagnosed patients. And you'll see the bandwidth of these squiggles to be quite broad, which means that when a new patient is newly diagnosed, physicians are pretty good about agreeing on what they will use to treat that patient. And in fact, it's very good. But so we started with, as you can see, a little over 400 patients. And that of those 400 patients, there were 1,692 treatments. That means that we were treating, these patients had received first line of treatment, progressed, second line of treatment, progressed, et cetera. So by this population had a median of 4 lines of treatment. What I'm telling you is that we had a good concurrence in the beginning. By line 4, not one patient was similar to the next. That's the diversity that we're dealing with. And if you want to learn from someone, you're looking for commonality so that you can form these cohorts. But this is the challenge we're dealing with. And this is a fairly structured way of treating people. But what it's saying is, number one, we have to get our act together and with guidelines and why we're treating the way we are. And two, no one institution, I don't care how big you are, has enough patients to do everything we need to do. So it's going to take this willing to aggregate, share data, and then performance analyses like this. But this, when I saw this, I was, oh my goodness. I knew it was going to be a challenge, but this is a major challenge. So that, again, reinforcement learning, the concept is beautiful, and through action and reward. I'm emphasizing that because when you want to inform that doctor in Kissimmee, Florida, who doesn't have a degree in genetics, but he's seeing a patient, or she puts that patient into our system, we need to be able to tell that physician, this is the population that looks like your patient, and here's what the treatments are. So it's that reinforcement learning that will be the background or the backbone for clinical decision support. All right, just again briefly, what's our foundation look like? How do we do this? Again, this is everything I'm about to tell you was created by M2Gen to serve the Orion members. When we built the first protocol at Moffitt, we used a SQL database, an Oracle, which was fine for one institution. When you bring 20 institutions, all with different types of patients and questions, but you need to harmonize it, a SQL database isn't going to work. So we went to the cloud, and the primary data sources are clinical, hopefully longitudinal, molecular, and because we considered the patient self-reported. So 3 extremely valuable sources of data. And what we've been able to do is annotate all of this and so that when you're talking about a patient, you know you've got different, or the types of data that you're looking for, okay? And we can actually do that within now seconds, depending on the population and question you're asking. And I'm going to give you some examples of that. This is the ecosystem for information. It is a cloud-based system, chiefly by AWS or Amazon. And you can see we partner with Hudson Alpha and TGen, 2 very high-profile laboratories. By the way, they had to agree to do the same platform, same extraction of the nucleic acids, so we harmonized it between 2 labs, which you almost never see. Even the TCGA couldn't do that. So, but to play with us, we told them, you're going to use the same format so that we can bring the data together and compare it. Once it's performed, it's hosted in DNA Nexus, where the pipeline and QC of the molecular data is held. Cloudicity is a hosting system for clinical data. It's HIPAA compliant and FISMA moderate, which is the highest level of security that you can get to date. And obviously security is extremely important, especially when we've got the active military in our system, right? And the visualization currently is ClickSense and cBioPortal. Those bioinformaticians in the room all know about cBioPortal. It's a great tool, but it only looks at about 30 clinical data elements. So we need more than one visualization of that data. This is just to emphasize that we take our security extremely seriously. Everybody that utilizes the data has to sign a data use agreement. In other words, they are using the data for a specific project, which the patients have been informed that's the way the data is going to be used. You violate that data use agreement, you're out, and probably more problems than that. But so everybody understands how this data is going to be used, because this is what we're telling patients how we're using their data. Okay, now, how am I doing with time? Good. As I mentioned, and you've heard me speak to this, we're here to identify patient need and create options to meet that need through anticipation. And we believe the population at greatest risk are those patients with more advanced disease that are likely to progress on standard of care. Hence, how do we use this system for clinical trials? And what you're going to see, I hope, is that we're going to use this total cancer care protocol as our prospective cohort surveillance, which is real time. It's refreshed every 3 to 6 months. And a physician can actually query it and say, my patient has progressed today. What were the options? And those options were already known before. And then it becomes a dialogue between the patient and physician about what's best for them. That's where we're headed. But what I'm going to emphasize now is the ability to determine through in silico design, how do you write a trial, who are you writing that trial for, in other words, what type of patients, and how do you find the patients for that trial. So consider it sort of an equation. We look at pharma or investigators, including our academic members, have a trial, they're looking for patients. We can tell them literally within minutes, this is the population and here's where they reside. For sponsor, has trial, looking for patients. But we also have patient at point of care who has now progressed, looking for trial. I will submit to you, working a patient up to determine what they're eligible for at the time of relapse is not the time to be working them up. That's the message I hope you're getting. We need to do this a priori. And that is the effort. And I'm going to give you examples and proof of concept of how it's working. All right, again, I don't have to tell you the challenges. You know what trials are. And this is a tough study that was done a little over 10 years ago. Each new therapy cost an average of $2.6 billion. That's 10 years ago, folks, so it's more than that. And it was taking 10 to 15 years to get from point of discovery to actually in the armamentarium. That's awful. What if, again, we could anticipate, not only populate the trial, but design the trial based on the data that we've created and gain go, no-go decisions much more rapidly. Not only is that better for the patients, but that's going to reduce the cost of doing trials. All right, and then we sit down with pharma and say, all right, we reduced your cost, ante up. All right, so again, just diagrammatically, in creating options in anticipation. Begins with the consent. That patient is then donating their clinical data and tissue for analysis. You saw the type of molecular data we're generating to do that, so it's very comprehensive, both DNA and RNA. And by the way, the Orion members are coming together about what's the next omic, and it'll probably be circulating tumor DNA and circulating markers, because we're going to find who's at risk sooner. That's the way the Orion members are thinking right now, but I hope proteomics gets included fairly soon in all of this. And so this is an ongoing approach of how do we integrate new technology in our assessment of the patient. But again, by following these patients, we create options for them in anticipation of relapse. So that is the process. Now this is an example of how we use it. CD30, in this case drug conjugate, is a target in Hodgkin's lymphoma, in some cases non-Hodgkin's lymphoma. And the CD30 is a very strong predictor of treatment to a monoclonal antibody against CD30. So Takeda, which has this drug with brentuximab, was thinking, okay, what's the expression of CD30 in other cancers, and will we have a population in need with solid tumors that could benefit from this therapy? So they came and asked us. We used the gene expression data that we'd collected on literally thousands and thousands of patients across all diseases, and did an H-index using R script. And we were able to predict, and this paper was published in JMIR, I think in 2014. And we were able to show that we could enrich populations with 75% certainty and predicting whether they would be positive for the immunohistochemical test measuring the protein CD30. So we found that population. Now, as we sat down with them, we looked at other aspects of solid tumors. Even though there were some expressors of CD30, they had other systems that would predict lack of response. So we were using the aggregate of data, and they decided not to pursue this, at least now. So using the data, early, if it's a no-go, you want to know it's a no-go sooner than later, right? That's important. So another example. This is also a antibody drug conjugate, but not against CD30. This was against guanylyl cyclase C. And the same question was asked by the sponsor, in this case Takeda, for gastric and pancreatic cancer. How many patients would express guanylyl cyclase C? And again, they had an antibody for immunohistochemical testing. So we were able to use the expression profile and looking at the population. So at the time they ask us how many patients in the total cancer care database have pancreatic and or gastric cancer. Well, at the time of this analysis, it was 6,564. How many would be eligible based on the inclusion-exclusion criteria for this study? We're now down to 440. How many did we pre-screen for using gene expression profiling? And you can see here, 269 for the IHC. So we took that 269, sent it to their companion diagnostic laboratory. They were blown away. We should have given them a heads up. We're going to send you 269 specimens. Because what they were used to was one at a time. The doctor in the clinic sees a patient. The patient's progressed with pancreatic gastric cancer. They think this trial might, if they know about it, might be useful, so they send in the specimen one at a time. No wonder it takes so long. We sent in 269 specimens for this test based on the data that we had, and found that 196 were positive. Now, none of those patients, the 196, needed the trial at the time of that analysis. Within the ensuing 6 months, 20 patients progressed and went on trial at one institution. That was Moffitt, because that's where this population came from. So Moffitt was one of 7 centers, major centers, participating in this trial. They enrolled 20 patients in the first 6 months. By comparison, the other 6 centers that did it by the standard measure one at a time enrolled a total of 9. 6 centers enrolled 9. The center in anticipation of the need and doing the assays before they need it enrolls 20. So obviously we're pleased to see that another proof of concept. It can work. Now, this is an abbreviated table of the challenges and solutions that we published in Clinical Pharmacology and Therapeutics about a year ago. I'm highlighting the major 4. I guarantee you the table is much larger than this, but I wanted to highlight some of the challenges we're dealing with. We've already talked about accurately defining a patient's phenotype. That means getting the clinical data and validating it. Got it. Security, you don't want to be over-secure though. If we create such restrictions that no one can see the data, that blocks the entire intent. Communicate with the patients you've consented. We've heard this loud and clear. So we do that. We let them know. We've studied. Now what patients told us is if you study my tumor and it's negative and I'm not eligible, don't tell me that. Tell me if it's positive and I can benefit. But I don't need to hear, number one, I don't want to give you permission every time you want to study it. That's number one. And number two, if it's negative, don't tell me. If it's positive and it might help me, that's when you tell me. That's what we learned from this advisory group. Okay, and ultimately, Orion's one network. If we're really to have an impact, we need to figure out a way of other networks combining forces using normalized approaches, including Common Data Dictionary. So those are the 4 challenges I would highlight. And because of time, I'm going to move forward. We have moved strongly. The Orion network has formed its own clinical trial network. And in so doing, we've created a single master trial agreement. We have the ability, as you've seen, we can identify the patients at risk and populate that trial and matching. We have a single scientific review committee, which the NCI mandates. If you're going to be an NCI designated center, you have to have an SRC. This is the first time the NCI has said you may select one SRC for 20 centers, providing it's an Orion trial. That was a big deal. Single IRB, and ultimately single trial budget. That is allowing us to open these trials much faster throughout the network because of the members agreeing this is what we're going to do to harmonize everything. If you had to have every one of them get their own IRB approval, et cetera, it would take forever. That's not here. So this is where we stand. We've got 7 trials that have been launched, including investigator-initiated trials, which is a question I've been asked by you, and I think that's fabulous. So yes. By the way, we consider every trial. And investigator-initiated will actually help you in terms of populating that trial through the membership. And we did that with a PARP inhibitor that started at University of New Mexico. It was an excellent trial, but they couldn't get enough patients. We opened it up to 7 other Orion members that wanted to participate and completed the trial in less than a year. And the power of the collaboration. And finally, you all heard about real-world data. We are meeting with the FDA frequently. In fact, I have a call next week. They want to know how they can use, working with us, the Total Cancer Care Protocol as an observational study for real-world data to generate real-world evidence, including being a in silico or synthetic comparator cohort. This is for phase 2 trials where the sponsor is looking for accelerated approval. They have to have some comparison. We become that comparison because, again, we have formed that cohort, right? You have the primary cohort, and there's a cohort that is eligible for a given trial. Those that go on trial obviously go on trial. Those that don't now become a comparison. The FDA is very excited about this. Now we have to prove it. So we can't just say we can do it. We have to prove that it's of value. And that's where we're in right now. But this is what I'm excited about, amongst other things. But this will, I think, accelerate the clinical trial process enormously. I just want to highlight again, we do remember the 3 categories of discovery, translation, which includes design of trials, matching patients to trials, and post-market. This is a post-market analysis for Revlimid or lenalidomide that Celgene has made for several years for myelodysplastic syndrome and myeloma. A study came out of Italy about 3 or 4 years ago, that suggested that patients on lenalidomide had increased risk of secondary AML if you had treatment for myelodysplastic syndrome and myeloma. As you imagine, that got the interest of FDA. Celgene came to us and said, and that was a population of several hundred patients. It came to our system. We had thousands of patients per category in myeloma and MDS. And we were able to do cluster cohort analysis of those that looked similar but had received lenalidomide and those that hadn't, and we showed no increase in risk. The FDA accepted that data, and we published the papers. So again, the power of comparator cohorts, including post-market. And again, this is something we can probably do in a second call where we actually go into the database, but this is the type of thing you would see. In this case, it's immunotherapy. And I just, by the way, asked my colleagues last Friday if they would do this for me, and they gave me the data on Monday. I said, I want to show them immunotherapy and what patients have it and what we're doing with it. So in less than a day, they had shown me the population's number of patients that had received immunotherapy, which were 27 different, so 662 patients with 27 types of cancer who had received a total of 129 different medications. Again, they did this in a couple hours. And they said as well, again, this is just to show you the unique, they itemized the number of patients on the unique medications, which were IO. As you can see, the top 9, I believe, and with each number. And then they said, well, let's do a subset analysis of that. And I will go, they did a subset analysis, and they said, we'll only show you the data, because it makes it easier to show, of patients where we're testing their primary tumor, not their metastatic tumor. It just cleaned up the data for demonstration. You can see the percentage of patients who had received immunotherapy, not surprising, kidney cancer, lung cancer were the top. And then I won't go through that. But this is the responders, non-responders. Again, this is done within an hour. They found 184 grand total who had received the therapy and had either response or non-response, 71 versus 116 responders across all those disease types that you see there. And then they did a Kaplan-Meier on responder, non-responder, and you're seeing it. What was used to take us months, they did in a few hours. So that's the data system. And they gave me all the mutations of all the patients that were in the system that met these criteria. So I saw 184 patients with their targeted mutation analysis and what was the indicator. And you can see those here. So I guess what lessons learned is we're getting there. If there's one word that I have to use to encourage you is perseverance. You're not going to get it right the first time, and this is a school of hard knocks. But I will say we're getting there. This is ultimately where we want to be, is at the point of care. This is the sine qua non. We know we've done it right when the doc in Kissimmee, Florida, can put the patient in our system and get data on the patient population that that physician is seeing at that point in time and get advice. We did a pilot on this for geriatric patients at Moffitt, and I welcome you to look at this. The database created options for each patient that was being discussed in the molecular tumor board for geriatric patients. It was very gratifying to see. This is our patient advisory council. We meet monthly. I have never had to ask them, what do you think? They're going to tell me. And they told me they want their own portal where we can tell them, not only thank them for allowing us to follow them and learn from them, but this is what we've learned, and these are the kinds of questions you should be asking. It's taken us 2.5 years to build it. It'll be launched in 6 weeks. I'm thrilled about that. So hopefully I don't have to enumerate this. I think this is why we're here. It's for the benefit of patients ultimately. And thank you very much.
So I think we're going to have time for just a couple questions. So, you have a question? I think you're on mute.
You talk.
Well, thank you.
I actually have a lot of questions, but I'll try to prioritize. Solid tumor metastatic disease, I mean, it's hard to really follow the amount, the quality, the distribution, the
Right.
genetics of mets. So do you have thoughts about how you might incorporate that into your platform?
Oh, it's in the platform already. I just didn't show you examples. Hematologic malignancies, almost every patient has a sequential specimen where we can study tumor evolution and compare.
Breast,
Yeah, breast is more difficult, and all solid tumors. I think in that population of solid tumors, we have about 20% of the patients have subsequent specimens. We don't biopsy, what we study is in excess of what is used for clinical decision making. So patients aren't undergoing extra biopsies to participate in this. They also told us they don't want to do that. But to your point, absolutely. I'm very excited about, we've just discovered by one of our members, there are about 140 patients with melanoma that also have brain mets. So we have the primary and the brain met. And that's going to be a huge study. That's where we want to go.
one other quick question is, because the gene sequence data is so powerful,
Right.
we're talking about value here.
Right.
So how powerful is the extra omics
Ah.
over the gene sequence analysis?
Excellent question. And I'm not avoiding the question, but it depends on the question you're asking. For example, if you're looking for a fusion protein, sequencing won't help you. So RNA-seq, and there are a lot of, well, not a lot, more and more therapies that are targeting, fusions and deletions, right? And more fusions. And it's that population, such as NRAP for lung cancer, where there are medications to hit that target, that's a must. And we're getting that on every patient. The other thing I didn't emphasize is everything you see that I demonstrated was RUO, research use only. We have now gone to a CLIA-based that using our RUO data to design the CLIA, both exome sequencing and germline, as well as RNA-seq. So we'll be getting fusion analysis with that.
Very nice presentation. I came from pathology. I have two questions. One, how's this paid for? It's very exciting. The second question, what is the diversity of your patients enrolled in this? I don't know if you covered the first question.
Yeah, I'll do the second question first. The diversity is actually quite high, and we can send you that data. As I mentioned, we're coast to coast, and with so I can't remember. Obviously, Caucasians are still the highest percentage, but very close to that is Hispanic populations. But then we go into Hispanic derivation from Mexico, from Puerto Rico, Cuba, etc. So we can tell you that. And you saw New Mexico, Utah, and Colorado are in, and they're enrolling Native American population. Oklahoma as well. Our Asian community is represented chiefly by City of Hope and University of Southern California. So the diversity is critical, and frankly, that's one of the reasons I'm excited about being here. I think that would assist as well. As I mentioned, Morehouse is a member.
But how is it all paid for?
Okay. We launched the protocol in 2006, and we did so through a partnership with Merck. So what I'm telling you is pharma is sponsoring most, if not all, of this data that we're generating to date. Now, they don't own anything. What they get is a license, and after signing a data use agreement, they get a license to mine the data for discovery, but the data belongs to you. So that data that is generated, the patients that you enroll, that M2Gen creates for you, we give it back to you through the information system. That's yours. And you can do anything you want. There are no what I'm telling you is pharma puts no limitations on how we use our own data, including you don't need their permission for publications. You make intellectual property discoveries, that's yours. And we don't have to ask them anything. But in order to pay for this, frankly, we needed a sponsor who would do it, and that's pharma.
So Fred asked a very important question about funding. How sustainable? I have a couple questions. How sustainable is this as you move forward in the next 5 to 10 years? Are they still going to be willing partners, or you're going to have to go somewhere else? That's question one. You address consent issues. You have an internal Ryan consent group, and that's critical. I didn't hear anything about quality control measures. And
Quality control of what?
You're set up to, at some point, look at the epigenome, how this might impact, and when do you expect to do that? And then the last two, metabolic data, or metabolomic data, and then microbiome effects. Do you have plans for this?
Well, first of all, I have a rule. I can't count beyond 3, so you ask 5.
I do that.
The first was how sustainable is pharma?
Yeah, funding.
Yeah, funding. Actually, we started with Merck. We've worked with about 15 different pharma now, and I'm pleased to say they're still coming in. Now, we have to be aware of what their questions are. And frankly, what I've learned is you have to work with them. You can't just give them data. You have to sit down, and they need help in analyzing this data as well. So that's part of what M2Gen does for them. And we do it for members as well. We hold courses on how to use CBio Portal on this data, for example, for the members. Now, I can't speak, obviously, to the future of will we always be able to enroll pharma as sponsors. I think we'll always have some involvement, but ultimately, we need to be working with payers where we're generating the evidence of the value of the therapy, and we are having beginning discussions with payers, and different hospital systems that are both payer and provider have a very strong interest in this. And we think by showing evidence of value for which populations will be ultimately rewarding in many different ways, including financially. Now, we will have never charged the patient, and we will never charge the patient. That's not what we do. But we will provide evidence of what's working for whom to payers. Your
So the other was sort of related to Jan's question, omics, the genome.
Right.
Omics.
Yeah, those are debates that are ongoing, not debates, discussions amongst the members. We're taking our lead from them. What we're hearing is the ctDNA and is probably the next step that we're going to take into that. Epigenomics is absolutely imperative, and there's a strong movement to include acetylation and methylation studies, as well as now the microbiome is, we'll see. That will probably be, much of these omics will become targeted to specific cancers, not across everything. We wouldn't be able to afford that. And I think we need to start with where's the most bang for our buck, and where's the biggest question that microbiome and or metabolomics may be having. And so we're going to rely on the questions you ask us to determine what the next omic will be. Quality, right. Quality is a mantra. I can't quote all the different pipeline analyses that include the quality. The molecular quality, frankly, is fabulous. And we use things such as TCGA as a comparison. We're always right there with them. The quality of the specimen, we went from, again, we're using FFPE primarily, but when we launched Avatar, maybe 25% of the RNA was usable for RNA-seq. We're now at 85% because of a new protocol that Covaris, both TGen and Hudson Alpha have adopted. The protocol is called Covaris. And that has just been an incredible addition.
So you have SOPs across all the 20 members?
Oh, absolutely. And the clinical data, we have a very rigorous QC process on that. And actually, it's stepwise. So the member sends the data to us, and they do their QC. We then do QC, and invariably, we always find something. We go back to them with our changes, and then that's part of the reason it takes so long, to be frank. There are 3 steps in QC on the same patient of the same data. And I'm not sure that's the most advantageous, best way to do it, but that's the way it is right now.
Last question.
So I was curious about tumor heterogeneity, both for looking for samples within the tumor that might be predicted for relapse, also for normal tumor mixtures in your sample that
Right.
is your data, and then also using noise or variations between multiple samples that might be predictive for response to the anti-PD-1 therapeutics such as Keytruda.
Mm-hmm.
Looking at the sort of polyclonality or different antigens.
Right. So on the tumor heterogeneity, it again depends on the tumor type. We can do it much more easily with hematopoietic malignancies, and we still use chiefly fresh frozen material, and you can do flow sorting based on target and separate cells, and we do that chiefly for myeloma and other hematopoietic diseases. Solid tumors are more of a challenge. we are not, we have entertained amendments to the protocol for targeted biopsy because as I said, the protocol itself uses tissue in excess of what's needed for clinical care. What you just spoke to would require additional biopsies that would be informed by imaging most likely. And the group is coming together to do exactly that. But again, this is what Intugen will do, is build a foundation to respond to that kind of request. And those kind of requests are coming in. And what was the other question?
How would you identify tumors that might be responsive to the checkpoint Keytruda-like therapeutics?
Okay, well, I don't know if you saw the slide that shows the different assays we're performing, but we're using MSI, PD-1, PD-L1 measurements, and there's about 4 more that I can't remember that are also run on every IO patient. So it's actually, that is one reason pharma does come to us. And frankly, Merck told us, they were our first group, the pharma group we worked with. They used the data to discover indications they weren't considering, and especially, for example, in breast cancer. They found they were the only one that went in because we had the data that showed them that it was going to work, and they got FDA approval. So, yeah, let me make this suggestion. When we set up the webinar, if you would send to us exactly what you'd like to see, we'll show it to you.
Well, thank you so much.
Thank you.
Appreciate you.
Thank you. Good group. As they say, it takes a village.